This is the last set of notes on the Unit 7 DNA and RNA. This one is about mutations. So mutations are changes in the DNA that, and when you change the DNA, when you change the sequence of bases in DNA, you're going to change the protein produced, which will definitely have an effect, or usually have an effect, on a trait or an enzyme or whatever the protein is programmed to do. There are two main kinds of mutations. One kind is a gene mutation. The other kind is a chromosomal mutation. We'll talk about each one. Gene mutations are often called point mutations because the change occurs at a single point in the DNA. This includes substitutions, deletions, or insertions of one or a few bases. Um, Substitutions sometimes don't even cause a change because you know that most amino acids have more than one codon that codes for them, and they usually only differ by about one base. And so if you change one base in the DNA, chances are it may not change anything at all as long as you're substituting it. But when you have a deletion or insertion, this causes what's called a frame shift, or a shift in the reading frame of the genetic message. As you're reading the message, the codons for the proteins, they're read in groups of three. And if you add a base in there or delete one, it's going to shift everything from that point on, one direction or the other, and change the amino acid sequence. Here we have a couple of examples of gene mutations. The original DNA here has GCA, which means the messenger RNA codon will be CGU, which codes for the amino acid arginine. If this cytosine is changed to a thymine in the DNA, that changes the messenger RNA codon to CAU instead of CGU, and this codes for the amino acid histidine. So it's going to change one protein or one amino acid in this protein. If it managed to change on to a base that gives a codon that is for the same amino acid, then it may not have any change in the protein. This might have a small change that might not be noticeable, or it might create a big change. The mutation in the hemoglobin gene that produces hemoglobin of one base, a substitution of one base, changes the protein so that the hemoglobin, rather than being normal hemoglobin, will be sickle cell hemoglobin. Sickle cell hemoglobin can be very problematic for the person who has it because it changes the way that hemoglobin functions in terms of carrying oxygen much less efficiently, and also changes the shape of the red blood cells so that they get stuck in capillaries. Here's an example of an insertion. Notice between the original DNA here and the mutated DNA here, you see that a thymine has been inserted between this adenine and cytosine. That changes all of the amino acids in the sequence. Insertions and deletions generally affect everything following the change, because the reading frame will either be shifted to the left or to the right there, and many, if not most, of the codons will be translated and transcribed wrong, giving the entire protein to be making the entire protein faulty. And it can cause any number of things besides just a problem somewhere. It could even cause death depending on what the change is. This shows a couple of examples. This picture is from your book. It shows you a normal gene here. And you see the sequence of methionine, lysine, phenylalanine, glycine, and alanine. And here, when we substitute, that's going to change just this one amino acid in the chain of protein. If you delete one of these uracils here in the messenger RNA, then that's going to change everything from that point on. Same thing if you insert one here, it's also going to change everything from that point on. So it can have a major effect on the transcription and translation of a protein from the faulty message. The second kind of mutation is a chromosomal mutation. This is a change in the number or structure of the chromosome. This includes 4 different kinds of changes: deletions, duplications, inversions, and translocations. In a deletion of gene, which is more than one base, it's a large portion of the chromosome is actually deleted during the process of producing the gametes, or during the process of cell division. And so you have a chromosome now that has fewer genes. It's a smaller chromosome, and there's definitely going to be a lot of things that are missing there. An inversion is when piece of the DNA breaks off and reinserts backwards so that the codons that are present in that region of the chromosome will be read the wrong direction, producing vastly different proteins. A duplication occurs between homologous chromosomes. These are pairs of chromosomes where you get in your body cells, you have one chromosome from mom and one from dad for each of the pairs of chromosomes that you have. So during a process here, you have the homologous chromosomes, a piece of one breaks off and adds to the other one, and so you end up with one that is shorter and one that is longer, having two copies of the gene, one with no copies of the gene. And then we have a reciprocal translocation that occurs between two non-homologous chromosomes, where a piece of one breaks off and attaches to the other. And this can happen during various kinds of cell division. As you can imagine, all of these mutations in chromosomes can have a very definite problem. And we'll talk more about this in our next unit when we talk about the production of the sex cells or gametes and the various kinds of problems that can result when things go wrong. This concludes this section of notes about mutations, and we'll see you in class.